All right, welcome guys to one of these shorter videos that we are going to have. This is going to be a very brief discussion of anaerobic respiration. And what I think that's Jeff Bezos or someone is saying, you got to spend ATP to make ATP is absolutely true. So, helps if I can move to the next slide. There we go. So every single cell on the planet, if it is alive, if it is made of cells, or if it is a cell, every single cell has to perform some version of cellular respiration. We can use cellular respiration as a general blanket term to talk about any means of breaking down organic molecules, extracting their energy and storing it in the form of ATP. So for all of these, you've got some sort of chemical energy in a molecule, and you're going to take that energy and transfer it into your handy dandy ATP molecule. Now, all cells on the planet can perform an anaerobic version of respiration, meaning without oxygen. And us as eukaryotes are very special. We can perform a more efficient version, which is aerobic respiration, which uses oxygen, which is why all eukaryotes on the planet do need to take in oxygen. And one thing I want to point out, animals, we're all eukaryotes. Probably pretty easy to remember. That means that we perform aerobic respiration and that we need oxygen, which is why we breathe it in. Plants are also eukaryotic. Anything that's multicellular is eukaryotic, which means that plants also perform cell respiration and they perform aerobic respiration, which means they need to take in oxygen as well. The thing with plants, though, is they also produce oxygen, and they tend to produce a lot more oxygen than they consume. Fungi are also eukaryotes. They're multicellular. You can see them with the naked eye. Therefore, they also perform cellular respiration, which means they also need to take in oxygen. And then our final group of eukaryotes, protists, some of them are unicellular, some of them are multicellular, but they all perform aerobic respiration. And we'll talk a little bit more about that in our next lesson. For now, let's take a look at basically how your cells release this energy. So the energy that's stored in a glucose molecule, you could release in one burst. If you were able to set it on fire, that would release all of the energy that's stored in it as chemical energy, and you would get a flash of heat, perhaps light, maybe even some sound. However, that large explosion of heat is not very useful. You can't harness it easily to do work, and it ends up being kind of a waste of usable energy. A lot of it's going to turn into heat and not be usable for an organism or for any system to really do work. So, and this principle also applies in car engines. So for example, you don't take all of the gasoline in your gas tank and set it on fire at once. Instead, small amounts of it are ignited with oxygen, and that releases small amounts of energy that are that's able to basically drive your car forward. Similarly, glucose is catalyzed or broken down in a series of very small steps, each of which has a small activation energy, and each of which releases a small amount of chemical energy, like what you see here. So that chemical energy can then, since there's a smaller amount being released, can be coupled to the production of something like ATP, which requires an input of energy. The net result of this, at least for aerobic respiration, is going to be carbon dioxide and water. There are other byproducts that are produced in anaerobic respiration, and we'll talk about those in a moment. Any molecule that has a lot of carbon-carbon or carbon-hydrogen bonds is going to be containing a lot of energy. And so if you'll notice, some of the molecules that contain the most energy, lipids, have a lot of carbon-to-carbon and carbon-to-hydrogen bonds. So they are storing a lot of chemical potential energy. Now, we've already talked about in class a little bit how electrons are what are carrying the energy, so we need something that will transport these high energy electrons around. One of the things we also talked about is that high energy electrons get fed into the electron transport chain. Well, something needs to take them over to the electron transport chain. If nothing took them over, then the energy of the electrons might just be given to random molecules and would just heat up the cell. So you have these special molecules, they're electron carriers. They are also called, they're a type of coenzyme, and their job is to carry high energy electrons. They pick up and drop off these high energy electrons, and when they pick up and drop off these electrons, they're basically transferring energy. There are two that are involved with cellular respiration. The first one is NAD+, which in a certain form, when it's carrying energy, turns into NADH, and we'll talk about how and why in a moment. And the second one is FAD. It is not necessary for you to know molecular details about these guys. It's not really necessary for you to even know what they stand for. If you're curious, you can always look it up in your textbook reading for Chapter 9, which you'll get soon. You'll have those abbreviations. The most important thing is to look at something like this. That equation is showing what ends up happening. So you have NAD+ combining with a proton and two electrons, and that forms the molecule NADH. NADH is carrying high energy electrons and is therefore able to give those to something like the electron transport chain, so that energy can be used to make ATP. So when I say they're electron carriers, you can think of them like a moving van, or if you want, you can think of them like an Uber driver. They pick up electrons, they drop them off. They pick up electrons, they drop them off. In the process, they're not changed. And in order for them to be able to pick up fresh electrons, they need to drop off the ones they have. Similarly, if you had a moving van and it was completely full of stuff, would you be able to load it up with new stuff? No, you've got to get rid of what's in there first. Also, if you were an Uber driver, would you be able to pick up a new load of passengers if you still had the old ones in the car? No, because they're taking up all the space. So notice that these molecules, when they are carrying electrons, they change form. They become NADH and FADH2, and one way that I think of to kind of remember that these are the energy-carrying forms of the molecules is to think of the H as standing for high energy. So if you look at these two, NAD+ and NADH, the one that has more energy is the one that has the H, standing for high energy. So let's look at this fundamental process that happens in all cells on the planet. It's called glycolysis. Even the name tells you what it does. Glyco means sugar, and lysis means, I hope you remember, to break. It is literally breaking a molecule of sugar in half. This happens in the cytosol, or in the cytoplasm. Keep in mind, it has to happen in every single cell on the planet. Every single cell on the planet does not have mitochondria. The bacteria, all the prokaryotes, do not have mitochondria, but they still perform glycolysis, and that's because this process happens in the cytoplasm. Every cell on the planet has cytoplasm. So what you have going in is you have one glucose molecule going in. You have 2 ATP that you have to put in to jumpstart the process. It creates a phosphorylated intermediate, which you see right here. Remember when we were reading about enzymes, phosphorylated intermediates tend to be unstable. That instability allows the cell to use enzymes to break this glucose molecule down into 2 3-carbon sugars. Once it's in the form of this 3-carbon sugar right here, and this other molecule that's formed, the other 3-carbon sugar is going to be converted to this glyceraldehyde 3-phosphate. The enzymes are going to rearrange these molecules and rearrange the atoms within them. In the process, they're going to grab a high energy electron, and that's going to be given to an NAD+, turning it into an NADH. Then a phosphate, an inorganic phosphate, and some ADP is also going to be used, and this product is going to be phosphorylated, and then that phosphorylation is going to be used to make an ATP. So Keep in mind that this is happening 2 times because you're forming 2 molecules of glyceraldehyde 3-phosphate every time you perform glycolysis. So you start with 1 glucose, you're going to add in 2 ATP, and that's going to end up yielding, in the end, 2 molecules called pyruvate, which is another 3-carbon sugar, 2 NADHs, and 4 ATP. Now, you used 2 ATP to get this process started. You produce 4 ATP. That means you have a net gain of 2 ATP. At the end of this process, you're left with 2 more ATP than you started with. So glycolysis, every time a glucose molecule is broken down using glycolysis, it produces 2 ATP. It also makes 2 NADHs. Those NADHs are going to become very key. Remember what I said, an electron carrier has to drop off its electrons before it can pick up any new ones. What that means is ultimately this NADH needs to be recycled back into NAD+, so that way it can come back and pick up some more electrons. And that's kind of where the difference lies in these different forms of anaerobic respiration. That's also where a difference lies in the formation of carbon dioxide and so on. So let's take a look. By the way, the steps of glucose one by one, you do not need to know the names of any of the molecular intermediates. In fact, the only ones you need to know the names of are glucose, which you should already know, and pyruvate. That's it. You don't need to know all these other things, fructose, 1,6-bisphosphate, glyceraldehyde 3-phosphate. Those are not necessary. You also don't need to know the enzymes that are involved. I am going to include a link to a video that shows this process happening in all of its gory detail, including enzymes. You don't need to know the names of those. You don't need to know the details of those reactions. In fact, this diagram that you see right here is even too much information for what you need to know for the exam. So you need to know you start out with 1 molecule of glucose. You have 2 ATP molecules used in the process. That is going to form 2 3-carbon sugars. Those 2 3-carbon sugars are going to release some high-energy electrons that are going to be given to NADH. They also, in the process of changing those 3-carbon sugars to 2 molecules of pyruvate, you're going to produce 4 ATP. That's all you need to know. So the anaerobic part in this is referred to as fermentation. So anything that is performing anaerobic respiration, you can technically say is fermenting. Basically, what differs here is the fact that NADH has to give up those high energy electrons to something, and that electron acceptor is going to be different in the different methods of fermentation. So all of these are going to regenerate NAD+, which is what you need, so that way they can go back into glycolysis and pick up those high energy electrons. This does not make any additional ATP, so the only ATP you make from this are the 2 that are made from glycolysis. So the first type of fermentation we're going to look at is lactic acid fermentation. Basically, the NADH reduces a molecule called the pyruvate molecule that's produced at the end of glycolysis and creates lactic acid. And in the process, it's going to be recycled back into NAD+. Lactic acid, you may sound familiar, or lactate. In fact, if you're an athlete, you may have heard that when you're exercising quite a bit and your muscles get sore, it's because they're making lactic acid. So when you're exercising heavily, you may not be able to get quite as much oxygen back to your muscle cells as they need. And so in order to still provide some energy, they can actually go into anaerobic respiration and perform this lactic acid fermentation. When they do that, their byproduct, instead of being CO2 like normal cell respiration, is this lactic acid. And that lactic acid buildup is actually what's responsible for you being sore after you've exercised quite a bit. That's why it's ideal to do a cool down after you exercise heavily. It kind of encourages the process of your body taking that lactic acid, converting it back to pyruvate, and then feeding it into the aerobic parts of cell respiration. So what happens is you end up having a glucose molecule, you end up converting it into lactic acid as well as some ATP. Keep in mind that this is just glycolysis. This is not giving you any extra ATP. So these ATP that you make during glycolysis are the only ones that you get. The entire purpose of making lactic acid is just to get this NADH back into an NAD+. That's it. Alcoholic fermentation is a different type of fermentation. It is still going to go through the same process of glycolysis. That is the only place where you will generate ATP. The difference between this and lactic acid fermentation is what you're using as an electron acceptor. So NADH is going to give its high energy electrons to a molecule called acetaldehyde. And in the process, it is going to turn it into ethanol. If you're wondering, yes, sorry, cats on the computer are not good. And I don't know how to get that back to normal. There we go. My bad. My cat would apologize, but she's honestly happy that she got on the computer. All right. I don't remember what I was saying. Let's say, oh yeah, the ethanol that's produced is actually alcohol, the type of alcohol that people drink that's found in beer and alcoholic drinks. In fact, you'll notice that this occurs in yeast. And yeast are actually the organisms that are used to brew alcohol. They're performing fermentation, they're respiring or performing cellular respiration anaerobically, and in the process, their waste product is ethanol. So just like we breathe out carbon dioxide as a waste product of our cellular respiration, they release ethanol. So I do tell people when they're drinking alcohol, they're drinking yeast pee. Nice, nasty way to think of it so that you don't want it anymore. Anyway, so what happens is the pyruvate gets converted to this other molecule, acetaldehyde. You don't need to know its name. All you need to know is that it ends up becoming ethanol. And in the process of making ethanol, you're going to take your NADHs and you're going to turn them back into NAD pluses. So that way they can go and they can pick up more electrons from a glucose that's being split. Same thing. You only get energy from glycolysis, no other sources. So, as you may have seen when my cat decided to step on the keyboard, this is our final slide. Like I said, very short lesson, and I wish I could say the same for the next one. It's probably going to be a doozy.